All right, so today's lab, water transport in plants lab is what we're doing. Okay, so that one you should be able to find in Google Classroom. Purpose of this lab, guys, is we want to essentially investigate and prove what forces a plant uses in order to transport water and where those forces originate from. All right, now, we've already gone over this in class. We know that what provides the force to transport water? Osmosis. Okay, where does that force originate? In the leaves. What part of the leaf specifically? The spongy layer, right, okay? Because water goes into the spongy layer, then it evaporates out through the stomates, but what gets left behind in the spongy layer is salt. Okay, so what we're going to do in this lab is we're going to have some plants, and we are going to alter or manipulate those plants a little bit, not the plants themselves. We're going to manipulate the structure a little, in that we're modifying them, so they're missing some parts. Okay. We are going to, in this lab, cut off a leaf off of the plant with as much stem as we can keep on there. Okay. We're going to place it in dyed water and leave it in there over the weekend. You probably did this in like grade 5 with a stick of celery. Okay. We're going to do this with a real plant. Okay. So what we're doing in this lab is essentially we're eliminating the roots. By cutting them off, we eliminate them as a source of what? Force. Right? If the roots aren't there, the only place that the force can come from to move the water is from the leaves. Okay? That's what we're trying to do. So we're changing this up so that we're making it so that, hey, if this plant can still transport water after all of these things I do to it, after cutting these parts off, okay, then I know, oh wait, that sounds like something you might need. It's an if and then. Okay? Alright, so what we're trying to prove here, guys, is where does the force come from? How are we going to prove that? How are we going to know at the end of this lab whether or not the leaf is the source of the force that moves the water? Okay? That's what we're looking to prove here. So, over the course of the next few days, okay, so we'll start it this morning and then it'll run today, Saturday, Sunday, and then Monday morning we'll look at our plants again. We're also going to be recording the water level that they are sitting in. Okay? Part of this, there's essentially 2 responding variables here, okay, in this lab. We're actually observing more than one thing. We're going to take before and after pictures of our plant. Okay, because we want to see if there's evidence of the water moving into the plant. How are we going to know if it's moved into the plant? It'll change the plant's color. Okay, we're dyeing the water. The dye will move into the plant. It won't evaporate with the water, but it'll move into the plant. Okay, and the other thing is we're going to measure how much water we start with and how much water we end with. So there's 2 responding variables here. Okay, those are the 2 things that we are going to use to prove or measure that water has moved into the plant or through the plant. Well, the way we're going to set this up, we should minimize that as much as possible. And I'll explain that when I go through the procedure. But yeah, you're right, and we're taking that into account. Okay. Things that need to kind of stay the same, okay, you've got to think up. So you've got to think up your controlled variables. I would say anything that could affect how well a plant transports water, carries out photosynthesis, that kind of stuff, factors that affect plant growth, okay, those things would be things that should be controlled throughout the weekend. All right? Everybody follow me on that? Just so you know, the temperature in the school does not fluctuate that much when you're not here. Okay. All right. It's not supposed to be very nice this weekend. Like cloudy. Is that okay? Well, it's still light. Plants don't die because it's a cloudy day or there's no trees in Victoria, Seattle, okay, where it's like they get 30 days of sunshine a year. Okay, they still survive there. So, just things to consider. All right. So we kind of a little bit talked about our hypothesis, but I'll give you a few more tips, pointers here. Okay. In the if part of our hypothesis, we should talk about how a plant transports water, and we just went over that in fairly big detail. Okay? and describes the experiment. I haven't gone over the procedure yet, so you'll fill that in later. Okay, then okay, how will you know, or what evidence do you predict will occur? Okay, and for that, I mean, talk about the two responding variables and what's going to happen to them in the lab. Okay, that's what you should always kind of talk about in the then part of your hypothesis. Then, this is the change in the responding variable that will occur. Okay, you're predicting your results. Okay, so the procedure is going to go like this. We're going to go across the hall to the lab, and we're going to take 2 graduated cylinders out of the cupboard. You're going to need a 100 mil graduated cylinder, and you are going to need a 25 mil graduated cylinder. Those are the really small ones. Okay, one of the really big ones, one of the really small ones. Okay, the reason is that we can't actually, we want to have the plant sitting in the small graduated cylinder, but we can't measure the water level all the way to the top because the lines, the measurement lines or graduations, don't go all the way to the top. Okay, so what we're going to do is we're going to take the small cylinder, okay, and we're going to fill it from the big cylinder. Right? So we're going to mix 100 mils of water with about 4 drops of food color. Okay, maybe 5 drops, no more than 5. Okay, you don't need it to be super dark. Okay. Now, I have only put red and blue food coloring on the lab table. Okay, because I used to put the yellow and the green out there too, but people would use it. If you're wondering why that's bad, that's why I don't put the yellow and green out there anymore. Yeah. On Monday, when you look at your plant, what will you see? Nothing. Nothing. Your plant is green and yellow. Okay? Yeah. It was funny the first time, and then it was just tragic. Okay? So I don't do that anymore. You're going to use the red and the blue. Maybe 2 drops of red, 3 drops of blue. Makes a nice dark purple color. Okay? You'll be happy with that. Okay? So you're going to mix 100 mils in the 100 mil cylinder. You're going to pour that into the 25 mil cylinder until it's very close, but not all the way to the top. Okay? So the lines, the measurement lines, end about here. You're going to fill that graduated cylinder until it is very close to the top. Not quite all the way, because obviously you've got to have room to put the plant in and have water be displaced. Okay? Everyone follow me on that? It's better to have it not full enough, put the plant in and go, oh, I have a little bit more room. Okay? Hopefully we won't have to do that because it kind of messes it up. But, okay? As long as it's going to be fairly close to the top, not overflow, but also your plant stem is going to need to be long enough that as water evaporates, the plant stem stays in the water. So you need to make sure you cut off a fairly long piece of stem. I inevitably have people cut off like this much stem, and then by tomorrow morning, the leaf is out of the water. Okay? And then the experiment doesn't work. All right, so make sure you cut as much stem off as possible, put it in as far as possible. Okay, so you're going to mix 100 mils, you're going to pour into there. Now, we're going past the level we can measure. How will we know how much is in there? Right, go 100 minus whatever's left in here, and that'll be your starting volume of water, and you need to record that. Okay, that'll be your starting volume of water. Now, as Seamus pointed out, water could evaporate from places other than through the plant. Yes, it could, which is why, once you have this part done, where you've filled your graduated cylinder and you know what your starting volume is, you'll come up to the front of the lab and I will give you the finger off of a rubber glove. Okay? And you will place that finger from the rubber glove over the top of the graduated cylinder, and you'll poke a hole in it with your pen or pencil. Okay? Then you will get your plants. Okay? The plants are sitting in the sink right now. The sink is full of water. Okay? And the plants are in it. when we get our piece of plant, we need to make sure that we cut it submerged underwater. Okay, so it's going to look, so that's right, that's the mixing process. Okay, cutting the plant underwater. Okay, I took a picture of like it. Okay, so you're going to make sure that the plant is underwater, and you're going to cut it under the water and put your thumb over the end that you cut. And then you'll quickly go back to your lab station and put the thing through. Take your thumb off, put it through. We want as little exposure to the air as possible, because if we get too much air into the xylem, what will it disrupt? The hydrogen bonds, okay? And that will really slow the ability of your plant to transport water, okay? So we want as best as possible to limit that. So have everything ready, then get your plants, okay? Quickly go over, put it in there, and then you're pretty much set, okay? So your final setup should look, well, you probably won't have that many leaves, because I got the plants I got had really big leaves and not very much stem, so you'll probably just get one big leaf, okay? So your final thing is going to look kind of like that, okay? You're going to have the leaf sticking out of the rubber glove, okay, that's wrapped over the top of the graduated cylinder, and we'll give you a piece of masking tape to wrap around the graduated cylinder and write your name on, okay? That way, on Monday, we can tell who's who's, because there'll be this class and my period 4 class all sitting on the windowsill. Okay? So we need to make sure we can tell them all apart. Okay? Everybody with me there? So you're building that apparatus. It's not a difficult apparatus. Okay? You just have to be careful about cutting it and making sure you have everything ready and getting your correct starting volume of water. Okay? Those are kind of the keys. Okay. Now, obviously, for the analysis and interpretation, we can't do that today. Okay? We can't do that until we have observations, which we're not going to have until Monday. So the observations you're making today are starting volume of water, and take some before pictures of your plant with your phone. Right? And take like lots of them. Take some of the top of the leaf, the bottom of the leaf, okay, the stem, zoom in on things. Try and get as many pictures as possible, and then on Monday you want to replicate those same shots. Right? So you have a really good fair comparison of before and after, because those are going to be your observations that you're handing in. They're not going to be lab drawings. They're just going to be kind of before and after, starting volume and ending volumes of water. Are there any questions about what we're doing when we get across the hall? Okay, so setting this lab up should take us like 15 minutes tops. Okay, it should really go pretty quick. Then we can come back and you can have some time to work on the pre-lab part. Okay, please.
When measuring starting volume, are you just kind of like, because you said the measurement at a certain point, and you want us to go past that?
Yeah.
Sort of kind of like eyeballing it?
Yes, you're eyeballing it. Don't take it all the way to the top. Have enough room, kind of, estimate enough room to put the leaf in without making it overflow. Okay? And not so little that it'll get out of the water. Delicate. Go ahead. Let's say groups 2 or 3. Okay? There should be enough plants around for that. Okay? All right. Any other questions? Okay, let's head across the hall. Okay, so good question. There are 2 responding variables. Yes, I want them both listed and explained. Okay, so there's 1 manipulated variable, there's 2 responding variables, and as always 3 controlled variables, all of which need to be explained. Okay, and in that explanation you're talking about how, if it's manipulated, how it's being manipulated, what effect it'll have. Responding variable, how it's responding, how you're measuring it. Controlled variables, how is it being controlled? What effect would it have if it wasn't? Okay, that's what goes in those explanations. Now, further analysis and interpretation. Okay, question number 1 is really easy. All you're going to do is compare that initial amount of water that you recorded today with the amount of water that's left in the cylinder on Monday. Just subtract them. They don't have the difference. That's easy. Okay. Number 2, what evidence do you have that water followed the path discussed in class? Basically, you have to talk about where the dye appears in the leaf. Right? We kind of know where water is supposed to go in a plant, what tubes it's supposed to go to and where, okay? But you need to kind of talk about the evidence you have based on the location of dye in your plant.
For the problem, do we have to restate it or just...
Yes, restate it in the form question, yep. Okay, and then for question number 3, predict the effect of adding salt to the water. So if we did this same experiment, but instead of using tap water, we used ocean water, or we added salt to the water, okay, what would happen? How would the experiment change? Would we get different results? What would that look like? Okay, you need to talk about that and why. Okay, those are your 3 analysis questions. Conclusion, same as always, copy paste your hypothesis, explain your acceptance or not acceptance with your data, okay, and then 2 sources of error, and how to fix them. People are kind of chintzing out on the how to fix them here lately. Okay, sources of error and how to fix them. All right, any other questions on that? All right, you got a little bit of time to work here before I go over the other labs.